All right, let's talk about building a phylogenetic tree. I love solving these problems, especially like the one that we'll see at the end. They're fun little puzzles to me. So let's talk about first some of the data that we use and some of the information that we use to infer evolutionary relationships to build a phylogenetic tree, and then we'll go through a practice problem. So remember we talked about homologous structures, right? So homologous structures that are similar in related organisms, but take on a slightly different shape or a slightly different function based on the unique evolutionary pressures that that lineage went through, right? Natural selection shapes these structures based on the environmental pressures that that lineage or that group of organisms was facing, right? So all of these plants have leaves, but due to the pressures of natural selection, some plants, those leaves have changed shape to form spines, right? Because those spines were advantageous in that environment. Some plants, their leaves have been selected to look like petals because those bright red petals were long lasting and they're advantageous in that specific environment. Same basic structure, but used in new and different ways based on the environmental pressures that that group faced. Right? So like we used also the example of the forelimb of many mammals or even tetrapods in general have the same basic structure, right? Humerus, radius ulna, carpals, metacarpals, phalanges, but natural selection has altered and tinkered with that shape, with these bones based on the pressures of that environment. For example, our hands are not very good for swimming. They're not very effective limbs for swimming, right? But slight mutations arose, slight differences arose, and then natural selection selected for mutations and for traits in organisms that live in the water to develop their forelimb into a flipper, right? But we can tell that they're still related because we see these homologous structures, right? Here's the phalanges of a whale, they form the flipper. And we can see that they're very similar to the phalanges in us and cats and bats, right? So these shared characteristics that are shared due to common ancestry are one thing that we can use to infer evolutionary relationships. But things aren't always super straightforward because remember analogous structures, right? So convergent evolution, similar environments can select for similar traits, right? So back to our example of cactus and leaves, right? So the spines on a cactus are modified leaves. We see a very similar structure and a very similar shape arise in a distantly related group of plants called euphorbia, right? These two organisms don't share this structure of spines and the overall growth pattern because they share a recent common ancestor. They share those structures because they faced similar environments in which these structures were favored. So analogous structures can make building phylogenies a little tricky. If we assume that those similarities in structures are due to shared ancestry instead of shared environmental pressures. Okay, kind of the same thing, like example over here, with the analogous structures of the dorsal fin of dolphins and the dorsal fin of sharks. They look pretty similar, yeah? Both of them are contributing to the aquadynamics of these organisms, right? The ease in which they can move through water, but the evolutionary origin of those dorsal fins is different. The anatomical structures and the genetic origin of those dorsal fins is not the same in sharks as it is in dolphins. Okay, so this is an analogous structure that arose through similarities in environment, not due to shared common ancestry. So this can make it a little tricky when we are building phylogenies and how we infer evolutionary relationships. A lot of work has been done lately using molecular homologies, comparing DNA sequences, right? The sequences of A, T, C's, and G's in organisms. They might look at RNA sequences. There's also a lot of work looking at proteins and the amino acids that go into constructing different proteins. So remember, amino acids are the building blocks of proteins, right? So the idea is that if a specific protein or a specific gene, the more related these organisms are, the more similarities in the composition of that protein or that gene those organisms will share, and vice versa. The less related those organisms are, the less similarities in their genes and their protein structures those organisms will share. So here we're looking at the amino acid sequence for histone proteins. So histones are used to coil up DNA, to store DNA inside the nucleus of a cell. And we're looking at similarities in the amino acid sequence in humans, chimps, mouse, rats, and cows, right? In these places right here that are highlighted, those are locations where there's been a mutation in the DNA that was inherited then by the next group, right? So cows, rats, and mouse all share an A amino acid, arginine right here, right? But it looks like in the ape lineage, there was a mutation in the DNA that was inherited where humans and chimps now have a serine amino acid right here. That's what that S represents. So if we look at these two right here, humans and chimps, they are very similar. We don't see any differences in their structure of the sequence of amino acids of their histone proteins. But if we were to compare a human to a cow, there'd be significant differences between the sequence of amino acids in the human and cow histone protein. And this shows us then, this can tell us then that these lineages diverged further back in time. Humans and cows are less related because those lineages diverged further back in time than humans and chimps. So humans and chimps, there's no difference in our histone DNA, in our histone protein, because we haven't separated. Our evolutionary lineages are very close together. There hasn't been a lot of time since our separation from chimps. So the idea here is that the amount of homology, the amount of similarity between either nucleic acid sequence, DNA sequences, or protein sequences can be used to build a phylogenetic tree, with more closely related species having fewer differences and therefore would be closer together, would share a more recent common ancestor on a phylogeny. Another key component when we're building phylogenetic trees is to keep in mind this idea of parsimony, sometimes also called Occam's razor, where the idea of Occam's razor is that the simplest explanation is the most likely explanation. In phylogenetics, we call this idea parsimony. So the simplest answer is the best. So the key idea here, though, is that the smallest number of evolutionary changes is the most likely scenario, because evolutionary changes are relatively rare, and so it's more likely for one evolutionary change to occur, and then lineages that diverge from that common ancestor inherit that evolutionary change, versus an evolutionary change being inherited many times independently in each lineage. So for example, let's take the trait of having 4 limbs, being a tetrapod, right? Is it more likely that the amphibian lineage independently gained that trait of 4 limbs and the mammal lineage independently gained that trait of 4 limbs, and then separately a third evolutionary event occurred where the reptiles gained that trait of having 4 limbs, right? So 3 evolutionary changes? Or is it more likely that that trait of 4 limbs arose once and then was inherited by all of the lineages after that point? So 1 change is more likely than 3 unique independent changes, just by random chance alone, right? Because gene mutations are relatively rare, and the same type of mutation happening in 3 different lineages is incredibly rare, right? The probabilities of that are astronomically small. So it's more likely that that change arose once, and then it was inherited by all of those lineages that arose after that point. So we are going to practice using what's called cladistics to build a phylogenetic tree. So cladistics is this idea that we can infer the relationship of organisms based on the number of shared characteristics that they have. And it's assumed that the higher proportion of characteristics that organisms share, the more recently they diverged from that common ancestor. So we are going to use these characteristics right here: vertebra, jaws, tetrapod, meaning 4 limbs. Do they have hair and fur? And do they have a notochord? So these are physical characteristics. These are the lineages, the groups of organisms that we're going to look at: lancelet, mammals, lizards, lampreys, and bony fish. And the way we read these kind of tables is that typically there's a yes or no, 0, 1, minus positive. So negative or no or 0 means that they don't have that trait. Positive, yes, or 1 means that they do have that trait, right? So lancelets do not have vertebra, mammals do have vertebra. Okay? So we're going to walk through how to interpret this tree and how to build out a phylogeny using this sort of cladistics scenario. So when we are building phylogenies using this cladistics method of this table of who has a trait, who doesn't have a trait, we want to start with the group that has the least amount of shared characteristics compared to the other groups, because that will be our basal group, or sometimes it's kind of called, you might hear referenced kind of as an outgroup. So if we look at the table here of the traits, vertebra, jaws, tetrapods, hair, fur, and notochord, they all have notochord, okay? But what group doesn't have any of the other traits that some of the other groups have? What group only has the notochord? The lancelets, right? So we can then say that here's the base of our tree. The first group to branch off would be the lancelets. Okay, and because all organisms on this tree have a notochord, we can put the notochord trait down here at the root of our tree. So it would look like this. Put a dash there indicating a trait, and that's our notochord. Okay, then we kind of go, what's the group that's next that has just one more trait compared to the lancelets? Which group has the next fewest number of traits? So if I'm looking at this table, I see that the lampreys have a vertebra and notochord, but none of the other traits. So they have 2 out of these 5 traits. So the lampreys would be our next group then. So we have another split, and then we have lampreys. And the question is, do all other groups, do the lampreys, lizards, mammals, and bony fish, do they all have vertebra? Yes, they do. So where can we put that vertebra trait? You can put it right here. Vertebra. All right, then which group has just one more trait compared to the lampreys? So lampreys have 2 of these traits. Are there any of these groups that have 3? Okay, so I'm seeing the bony fish has vertebra, jaws, and notochord, but it does not have forelimbs or hair and fur. So that means that this group split off like this. Then we have the bony fish. Okay, and that trait that bony fish have, jaws, is that also shared by the remaining groups, the other groups that we haven't put on our tree yet, the lizards and the mammals? Yeah, it is. Okay, so it means that we can put that trait of jaws down at the root right here, down at that base. Right here, so jaws. So it means that everything past this point, everything beyond that point right there has jaws. Everything beyond this point right here has vertebra. Everything beyond this point right here has a notochord, right? Because it would have been inherited by the common ancestor. The common ancestor right here would have had jaws. And then our last 2 groups that we have left are mammals and lizards. So it doesn't really matter the order, again, because if we rotate on these nodes, it's showing the same relationship. So we have mammals and lizards. And do they both have, so mammals and lizards, there's 2 more traits that we haven't marked on here. We haven't marked on tetrapods or hair and fur. And where does it make sense to add these traits on? So we could put them both here, hair and forelimbs. Okay, so we could put 2 traits there. But do the lizards have hair? No. Well, we could do it this way. We could say the common ancestor had hair, and then lizards lost hair. Okay, so gaining and losing a trait can both be
something that we mark on these phylogenies. But is that parsimonious? Does that follow our rule of the simplest answer is the most likely? No. What's more likely? It's more likely that 4 limbs evolved here, and then hair evolved here. So instead of 3 evolutionary events, hair, 4 limbs, and loss of hair, we have only 2. 4 limbs evolved here, and then hair evolved here. So I hope this helps. We'll be practicing this in lab. But that's basically how you use a cladistics table to build out a phylogeny.